All right. Well, thank you all for coming, and thank you for the invitation to come and speak. I think it's amazing that we have a native bee society here in Washington State. Very excited to watch the society as it continues to grow and evolve. I think it's really really amazing. So, as Kate mentioned, I'm a professor at the University of Washington, and my work is on plant-pollinator interactions and global environmental change. So obviously, native bees are something that are very near and dear to my heart. All right, so today I'm going to be giving you a little introduction on pollination networks, what they are, and why they matter. And then talk about kind of go into a little bit of depth with two different sets of field experiments that we've run out of my lab. One that both of which relate back to this idea of global environmental change. So one is focused on what happens when we have pollinator extinctions in the system, and how does that matter in particular for plant reproduction. And then secondly, we'll get into some more recent work that we're doing on climate change, and in particular, timing mismatches between plants and pollinators, and how those timing mismatches, again, might impact pollination, so plant reproduction, as well as pollination network structure and multiple other facets of that system. And then I'll just talk a little bit. I have this labeled here as future work, but it's really sort of more about other work that we're doing. So just giving you very, very brief highlights on a couple of ongoing projects. So I'll get this kicked off with the network piece, and what are pollination networks, and why should we even be thinking about them? So in terms of this introduction, kind of getting into what networks are more generally, and specifically what pollination networks are, and then again, this sort of idea of why do we care? Why would we use these? So what are networks? I think this is an idea that all of you have heard of in some way or another. I think in particular, we tend to think of computer networks, social media, maybe IT type networks, things like that. And that certainly isn't wrong, but this concept is also really very relevant for many biological systems at multiple different levels of organization. So we can really create a network out of any sort of set of discrete entities that might interact with one another's, and we tend to call those nodes. So those could be computers, but they could be people, right? So we could look at a social network by connecting different people. But they could also be things like genes, for example. Gene regulatory networks are a really hot topic right now in molecular biology. And they could also be species, and that's what I'm going to focus on today. And we can start to build the connections between these nodes. And again, these connections can be all kinds of different things. So they could be physical contact between two species. They could be information signaling. It could be transmission of COVID or other pathogens, or they could be feeding interactions. And we can then begin to sort of scale up from small sets of nodes and their interactions to really understanding the connections between much larger sets of nodes in a wide range of different contexts. So one cool example from biology, and again, a very active area of research right now, is neuronal networks. So the connections between nerves, and this is a map that was published fairly recently of the neuronal network in this nematode, C. elegans, that's often used as a biological model system. But all of you aren't here for nematodes, you're here for pollinators. So that's what I'm going to focus on, which is pollination networks. And so we can begin to decipher these patterns of connections between plant species and pollinator species. So on the right, you'll see a very stylized network with 5 pollinator species and 5 plant species, and tracing out those connections. So that's what we're then really looking at the in terms of the real data that are on the left there. This is a pollination network from southern Spain that Jordi Bascomte's lab put together. And so that's really the basics there. We're just tracing the connections between the species that interact with one another. There's been a very little bit of work that's been done at levels other than the species level. So just some very introductory first steps into thinking about all of the different individuals, say, that comprise a species and where they're going at an individual level rather than at a species level. But almost all of the work is really at the species level, and that's where I'm going to stay for the rest of the talk. So why use networks? I think one of the questions I've heard, or really even criticisms I've heard, is, okay, well, you can create these kind of pretty network diagrams or interesting looking network diagrams, but is there anything more than that? Do these networks actually matter? And I think there are a lot of really cool examples of why these patterns of connections matter. But I think one of the most compelling comes from Washington State, but from a very different system, not from pollinators, but from intertidal organisms. And this is work that was done by Bob Payne, who was a professor at University of Washington for his entire career, and became really famous in the science world for an experiment that he published in 1966, using this set of interacting organisms in the intertidal environment, where the arrows that you see are connections, but they're feeding connections. So the starfish is actually feeding on all of these organisms in the system. And then there's also a snail predator that consumes a couple of those species. And so what Bob Payne did is he did removal experiments of these starfish, and sort of ask what would happen. And if we were to look at kind of isolate any one pair of species in this system, like the starfish and the limpet, you would basically expect that this would be a good thing for the limpet, right? Its predator has been removed, and so it's probably going to increase in abundance. But it turns out that is actually not the case. When you remove the starfish, all of the other species basically went extinct except for one. This mussel, this middleus, then competitively displaced all of the other species. So this is something that by looking at just one interaction, we really could not have predicted. And we need to really understand the network as a whole to get that systems level power to understand what's going on. And I think this is particularly important in the context of understanding how global change will impact organisms and ecosystems. And this is called the keystone species concept. So this experiment really is what gave us that idea of keystone species, and again, that made Bob Payne quite well known in the scientific community and really even in the broader public. So that's my introduction. So I'm going to switch gears here and start talking about some of the work that we have done and are doing in my lab, starting with work on local extinctions. And I think that this is a very, very salient concern in terms of native bees and other pollinators. So we can think about a local extinction as when a species is lost from a particular geographic place. And it need not be lost or going extinct globally, although that can be a concern. And this is Franklin's bumblebee that haven't been seen in a couple of decades now, that was once known only from parts of Northern California and Oregon. And it's thought to potentially be globally extinct, again, hasn't been seen in a couple of decades. But we have many other examples of native bees that have had major range contractions, so where they are no longer found in many of the places that they used to be. So these local extinctions really are a major concern when it comes to pollinators. And I think an important question that follows from that is, if we're losing pollinators, what is going to be the impact on plants? Because pollination is ultimately about plant reproduction and supporting reproduction of plants by fertilizing a plant's ovules that produces viable seeds, and then also allows fruits to grow. So obviously there's a connection to, very strong connection to agriculture there, and agricultural pollination, as I think probably all of you know. But in terms of this concern, I think one thing that potentially makes it less of a concern is that most plant species, when we actually look at them, when we trace out their pollination networks, most plants are visited by multiple pollinator species. And so they may actually have a little bit of built-in backup if we lose just one of those species. So this is work, the work I'm going to talk about is work that we did in and around the Rocky Mountain Biological Laboratory in Colorado. This has been our primary field site for about 13 years, and has been a really wonderful place to work, really beautiful place. When I lived in Atlanta, it was a great place to escape the heat and humidity of the summer and get up to Colorado. And now that I'm in Washington, I'm still continuing to work here in Colorado, but also expanding out to some more local field sites in Washington. And I'll mention that just a little bit closer to the end of the talk. But in particular, there are multiple bumblebee species at the Rocky Mountain Biolab. Depending on what you count as sort of an abundant species, we have 7 or 8 species that are relatively abundant there. And all of those species visit this beautiful purple larkspur flower, the tall larkspur, Delphinium barbii. And so the thought here was that if we take one of those species out, if we remove it from this system, really could we see functional impacts of that in terms of plant pollination and plant reproduction? And again, because this plant has a lot of other species that could potentially provide backup, would we see an effect even just by taking out one of those species? And again, in some ways very much motivated by multiple previous studies that had been done, including some bumblebee removal work, even starting in the 1970s by David Inouye at the Rocky Mountain Biological Lab, but also this work by Bob Payne, and really trying to say, well, we could expect that we might see backup, but we also find these unexpected effects when we do removals, or more generally when we mess with ecosystems in lots of different kinds of ways. So what will we actually see when we try this in the field? And in terms of predictions, I have this prediction that if we remove a species, so these pollinator species losses, would actually drive a reduction in what's called floral fidelity in the remaining pollinators in the system. And that loss of floral fidelity in turn would lead to reduced, or could lead to reduced plant reproduction. And I'll take just a step back here to say, in some ways, this is like the best kind of case scenario. So maybe the most conservative scenario, one of the most conservative scenarios of, if you were to remove a pollinator, what would happen to the plant? And so I'm not going to get into the sort of theoretical background for why we would expect to see this. I'm happy to talk about this with anyone who is particularly interested, maybe after the talk, but there is some ecological theory that leads to this prediction. So what is floral fidelity? I know many of you have had the pleasure of just watching pollinators visit flowers. And in particular, we can often see this behavior with honeybees and bumblebees and other very conspicuous pollinators, as well as many other species, but we often see them sequentially moving from one flower to another flower of the same species. And this is exactly what the plant wants from a pollination perspective. In so doing, they're transferring the right kind of pollen in between individuals of that species. By contrast, we don't always see this. Well, and before I move on, I'll say that this is um, a behavior that has been noticed by naturalists going back to Aristotle. Darwin wrote about it as well extensively. So this is something that actually has a very long and kind of fascinating history of the intellectual thought behind it as well. Um, so in contrast to this idea of floral fidelity, we often see a lack of fidelity, or we can see a lack of fidelity in some systems. And this is where we might see a pollinator moving between individuals of different plant species, so different flower types. And we know that lily pollen doesn't work to pollinate a rose, right? Plants need pollen from their own species in order to reproduce. So this is actually quite bad from the plant's perspective. So not only are they taking pollen away that could have been used for another plant, they're getting the wrong kind of pollen deposited on them. These pollinators are also drawing down the floral resources that could even attract other pollinators. So again, really not an ideal scenario from the plant perspective. But we really wanted, so this is the hypothesis here, that when we remove the most abundant single bumblebee species from the system, that we would see this shift in the remaining species. And then we set out to test that idea. So in order to do this, we had to remove the most abundant species of bumblebee. And this is work that we did in the field using targeted netting. So we basically just went out with nets and caught the most abundant bee. We worked in 20 by 20 meter plots. And we couldn't always get 100% of the individuals, but we really did a good job and on them for the most part, and kind of patrolled these sites to kind of make sure that there weren't other individuals of that species coming in. And I think typically what we were doing is sort of draining down most of the individuals in the nearest nesting site for that species. So we would work in a site and compare it in a control state, so without that removal, and then in a manipulated state with the removal. And we literally followed these bees around as they foraged to observe the floral fidelity. And then we also removed pollen from these bees actually in the field. And I should say we did all of this non-destructively. So we did it without killing the bumblebees that we were removing. We just captured them, kept them in vials, and then let them go, put them in a cooler, and let them go at the end of the day. So here are a couple of members of our team in our very first field site to Emory undergrads. Actually, on the left is Therese Lamperty, who's currently a postdoc in my lab. She went on to do a PhD at Rice and then circled back and is now doing a postdoc with me at UW, which has been a very fun turn of events for me to have in the lab. And like I said, we caught the bees, put them in vials, and then kept them in the cooler so we could let them go. And we have this kind of funny setup here so that we could do the removal of pollen of the bees. So these weren't for the removed bees, but ones that we were capturing that were in the non-removed species, so that we could look at their floral fidelity. And we would bring this table and microscope out to actually do that pollen removal. And then later we would look at the composition of pollen that the bees were carrying to see if they were really just focused on one plant species or if they were visiting more than one. And like I said, following bees around in the field, which is probably the most fun field work I've ever done in these beautiful wildflower, high elevation wildflower meadows in Colorado. And as I mentioned, our target plant is Delphinium barbii. It's in the buttercup family. I've got well, this is a picture of it here, and I've got a couple more coming. And it is the most commonly visited plant among all of the bumblebees in the system. And we did some work to really standardize this to make sure that we were really only, the flowers that we were working with were only available for a standardized set of 4 hours in either the control state or the manipulated state. And then we looked at those flowers in terms of what was the pollen that the bees deposited on them, and how much then, how many seeds did those plants produce. So here's the picture that went into the flyer, which shows one of our target plants. You can see there's a stake, a very thin wire stake just off to the left that allowed us to bag that flower without damaging it after our time was up. And then we had these particular flowers that we were focused on in terms of uh, looking at the pollen on the stigma and then actually tracing that all the way through to seed production. So really looking at all of those steps from observing the bumblebees and measuring their fidelity in the field, to looking at the pollen that they were carrying, to looking at the pollen that they deposited on flowers, to actually measuring seed set in those flowers. So we did this in 20 sites. We actually manipulated 6 of the 11 bumblebee species in the system. And again, like I said, there are really only 7 or 8 that are common. So we were really manipulating most of the different possible species that we could in this spot. And we recorded over 23,000 individual pollinator, individual bumblebee foraging movements as they move from one flower to another, counted almost 50,000 pollen grains, and also counted quite a lot of seeds as part of that work. And I'm not going to go into a bunch of graphs or statistics, but instead I'm just going to say, as we traced this through each of these steps, we indeed found exactly what we hypothesized, that there was a statistically significant difference, reduction in flora fidelity as we move from the control to that manipulated state. Similarly, bees were carrying more single species pollen loads in the control relative to the manipulated state, where they're more likely to be carrying 2 or more species. Similarly, with pollen deposition, we saw that more of the stigmas in our control had just the right kind of pollen, so pollen from other delphinium plants, whereas in the manipulation, the species removal manipulation, they were more likely to contain pollen from other species as well. And then finally, this translated to actually greater seed production in the control relative to the manipulation. So really kind of were able to trace that all the way through. And so ultimately these experiments show that we can see a significant negative effect of removing a species from the system, even though on the pollination of this target plant, even though the target plant has many other plant species, or pollinator species, I should say, that could serve as or serve as a means of redundancy there. And finally, while we did remove pollinator individuals, we were able to control for that statistically. So these differences that I've been talking about are not due to a lower overall abundance of pollinators in this system. And so this is work we published about 10 years ago in Proceedings of the National Academy of Sciences, which is fun with I published this with Heather Briggs, who had been a grad student at UC Santa Cruz, but I was on her committee, and she was very central in making this work happen. And we also followed up on this with some other collaborators to look at what happens to the entire pollination networks in this system. So not just the bumblebees, but also all of the other bee species and all of the other pollinating insects, flies and butterflies and many others. So what happens to the structure of those networks when we do these species removals? Don't have time to talk about that today, but again, I'd be happy to chat with anyone about that as well. So this is a good transition to thinking about the second kind of component here, which is thinking about climate change. So I will dive right into that. So first of all, you'll notice that the slides in this next section coming up have a very different sort of visual flair to them. And this is really work that was driven by my former PhD student, Sigmund Loy, who is now at the Atlanta Botanical Garden, and who is not only an amazing scientist, but also an amazing artist and graphic artist. He's produced a bunch of posters for the government of Singapore, where he's from originally, in addition to multiple other professional graphic art work that he has done. So Loy is really responsible not just for the science here, but also for the beautiful slides in this section. And this is also collaborative work done with a bunch of other folks, most of whom here are former members of my lab, Annie, Connor, and Victoria or Tori. And we also were inspired to do this work by Heidi Stelzer, who's on the faculty at Fort Lewis College in Colorado, and who also works at the Rocky Mountain Biolab. She did a very different kind of work, but is the one who kind of really helped to inspire the experimental approach that we ended up using. And this work is really motivated by the idea that with climate change, we're seeing changes in the timing of multiple different kinds of biological phenomena. So things like when migratory birds come through, when trees leaf out for the spring, when plants bloom, and when insects, including pollinators, emerge, often from the ground, to begin pollinating things. So, when we see this earlier flowering, and this is something that we are seeing in more and more places around the world, how is that impacting fecundity or reproduction in those plants, and specifically with regard to pollination? And we can see impacts of this earlier flowering in two general ways. So one is through environmental or abiotic effects. So this daffodil that you see here is, it bloomed and then it got snowed on, right? So if you bloom early, you're more likely to have these kinds of events happen to you, like a cold snap comes through, and probably any of you who are gardeners have experienced this kind of problem before. We can also have problems with mismatches, timing mismatches with mutualists, including pollinators. And it's this second part that we're really focused on. And our idea is that in our system in Colorado, when many of the plants there, their blooming is driven by snowmelt timing. So when there's earlier snowmelt, there's earlier flowering, and there's some really beautiful long-term data from the Rocky Mountain Biological Lab, actually going back almost 50 years now. Actually, I think this year it will be 50 years, on timing of bloom for multiple species, and so showing a really strong effect of snowmelt in terms of that bloom timing. So, so the idea is that we could potentially accelerate snowmelt to accelerate flowering, and really get at this question of how might these timing mismatches impact plant reproduction. And we're definitely not the first people to wonder about this question, or to even conduct work in this realm. There have been some really nice, really beautiful studies that have, for example, manipulated flowering time of potted plants, using, putting those potted plants in the greenhouse and basically forcing them to bloom, and then taking them back out to a field context. There's also been work that's used this idea of snowmelt acceleration or snow removal to speed up that timing of when plants bloom, and then looking at, if things bloom faster than expected, how does that impact their reproduction? So this is a very elegant approach, very nice manipulative experiments, and this work has shown some impacts of early bloom on plant reproduction. But at the same time, Really, all of this work has been in a single or maybe two species context at the most. And we know from many, many other kinds of studies that the context of who is around you and what they're doing is really important, and that plants wouldn't bloom alone in nature. Instead, if they're blooming earlier, they'd be likely to be blooming with multiple other plants. So then our idea became to accelerate the snowmelt, but rather than to do this with just a little snow removal manipulation of, maybe a few inches by a few inches to get one plant individual, then instead we do this in large plots. And really asking if this advancement of flowering that would lead to a timing mismatch might impact overall seed production when we look across all of the species, plant species in this in a site that would be pooled together. If it changes the reproduction of different species differently, and if those differences are due to really specifically due to pollen limitation rather than any changes in, for example, water balance or other things that we might change by doing these snowmelt acceleration experiments. So again, wanting to do this in big plots. And so this is our design. We worked in these 10 meter by 14 meter plots. So really about the size of a tennis court, more or less, with somewhere around 12 meters between a control plot where we didn't accelerate the snow and an experimental manipulation plot where we did. And there's a picture up in the top right of one of these plots being deployed over the snow. And what we did to melt the snow is put a big piece of greenhouse shade cloth, that black fabric that you see in both the schematic and in the photo, over the snow. The dark color absorbs sunlight and really heats up that fabric and thus melts the snow faster. By doing this, we can advance flowering by 1 to 3 weeks. And so this is the snowmelt treatment again on the right, where we have it melted out earlier than the control and stuff starts to bloom. The very first set of species begin to bloom in there before anything is going on around it. And then similarly, as we look out to other, as the season progresses, the rest of the snow will melt, and the control plot basically looks like the surrounding area in terms of what's in bloom, whereas the snowmelt plot now has different things blooming in it. So the snowmelt acceleration impacts the timing of bloom, not just for things that bloom right when the snow melts, but even for things that are blooming much later in the season. So this is our design in terms of a single site, and then we replicated these sites in 8 different places. So each one had a control and a manipulation plot. And we tracked the phenology, which is the timing, and specifically the flowering phenology, and then also the reproduction of 13 different plant species, representing about 1,100 individuals. And here's Loy in the field with our other former student and then subsequently former lab manager now, Annie Schiffer, who's doing a PhD now at the University of Utah, so right near the Logan B Lab, which is fun. So here are those 13 species. There's actually a different species of delphinium, a smaller one that blooms earlier in the season, Delphinium natalianum, along with several other beautiful wildflower species. And I mentioned that we also wanted to know if these effects were driven by pollen limitation, if we see any effects if they're driven by pollen limitation, as opposed to resource limitation, for example, due to water dynamics. And in order to determine this, we also did pollen supplementation experiments, also known as hand pollination experiments, to really get at this idea of pollen limitation in 9 additional species, although the data unfortunately only really ended up being usable for 5 of those species. And here you see again Annie and Loy painstakingly hand pollinating flowers. So this is just really a crushing amount of field work, largely driven by the two of them for this piece of the puzzle. So I'm going to jump right into some results here. So our snowmelt accelerations worked. So here's one picture of an accelerated plot with the control adjacent to it. You can see, especially on the left-hand side of that plot, that it's really started to melt out much faster than the surrounding area. And in particular, here's a graph where we have the control in the colder blue and the accelerated in the warmer red. And we can see that that's shifting the date of snowmelt to earlier in the season for all except for one of our plots. And indeed, this also led to earlier flowering. So I've got the sort of plot area covered, but if we remove that, we can see some beautiful glacier lilies and spring beauties, Claytonia, in the plot there that are in bloom, whereas there's much less in bloom around that. We also did advance the timing of flowering. This plot is a little hard to read, but as you move toward the left, that's earlier blooming, and this is all relative to the control, which is that vertical line. And unfortunately, this worked in 6 of the 8 sites, so we were only really able to use those 6 sites. But that is really how it goes very often with field work. So again, circling back to the questions that I mentioned before, so how is this advancement of flowering impacting overall reproduction pooled across the community, species-specific reproduction, and pollen limitation? So in terms of pooling the data across species and sites, we can see a statistically significant negative effect of these timing mismatches on plant reproduction. So this is striking, I think, result and really pointing toward this really not being good overall for plants and for plant reproduction in particular. And then we can ask, does this effect differ from species to species? I basically have the same kind of plot here where, again, the blue is the control and the red is the accelerated. So if the red is lower than the blue, that was bad for the plant. And that's what we see in both of these two species. The furthest left on the graph, Bucra stricta and Delphinium natalianum. This Lathyrus, however, on the other hand, the one that's third over, actually we see a positive effect of this accelerated bloom on its reproduction, whereas for most of the others, they're maybe a little bit negative or essentially flat. So this really is impacting different species differently, so potentially creating some winners and some losers. We also found impacts that these impacts are being driven by pollen limitation. I'm not going to go into the actual graphs because they get very complicated to interpret, and it takes a little bit of setup to even sort of walk folks through that interpretation. So I'm just going to leave it that we did in fact find that these effects were due to pollen limitation rather than to resource limitation. So really seeing again in terms of all 3 of these questions, yes, impacts on overall fecundity and negative impacts. We see species-specific trends where there are winners and losers, and we also see that that these trends are being driven by very likely by interactions with pollinators rather than anything else. So there's a lot of follow-up work to do on this. I'll say that this paper is now in revision at a journal called Ecology Letters, but a lot of really fun ideas for follow-up work that we would really like to do. One of the challenging pieces about this is we were able to do this work 1 year and then tried to repeat it multiple other years, but have been really hammered by the mega drought in Colorado. So if there's not enough snow, it's really hard to accelerate snowmelt timing. And so that makes things challenging. So this is actually work that we are hoping to start working, not just hoping, but planning to start working on in the Washington Cascades. And we've just secured a pilot grant from the UW program on climate change to actually do that piloting of doing this work in Washington. But yeah, we're interested in many other things. So that this includes the pollinator side of the equation, which I know all of you are very interested in. But in particular, we're doing some analyses right now because we do have data on the pollinators that we're visiting these plants, really to see if there are timing mismatches with the pollinators, and if those are related to if those timing mismatches are directly related to plant reproduction. Again, we have some indication that they are, but that's from using the plant side of the equation only, and both the plant and the pollinator data. When we started this work, we were very focused and curious on how this might impact pollination networks and the structure of those networks. It turns out one field season of doing this is just not enough to pull out any trends there. It's very challenging to sample these networks well. So if you have 10 plants and 10 pollinators, that's 100 potential interactions to observe. In reality, no pollination network is as small as 10 by 10, at least not to our knowledge. So even some of the smallest ones that have been looked at are larger than that. So with more than 100 possible interactions to have to characterize. So it really takes a lot of sampling in order to make this work. So we're excited to kind of try to think about ways that we can get multiple years of data here. And again, hoping to do that by moving to the Washington Cascades. So yeah, I just wanted to very briefly mention a couple of other projects I'm involved with, and I realized I'm missing a slide here on the Meadow Watch program. This is a really cool program that was started by Janneke Hillary-Slambers, who was at University of Washington and actually moved to ETH in Zurich, Switzerland, at the same time that I came to UW, which I was disappointed about. But Meadow Watch is a really fun citizen science project that tracks the timing of when plants bloom in Mount Rainier National Park. So obviously very relevant to the last project that I mentioned. And we're always looking for volunteers to go out and hike the trails, beautiful trails in Mount Rainier, and help us collect data on which species are in bloom at which points along these trails. And actually, I'll just go ahead and put our website into the chat if anyone is interested. So I just did that. So that's one of the other projects we've got up our sleeve. I'm also doing some really fun work with two collaborators, Fernanda Valdovinos, who is at UC Davis, and Mark Novak at Oregon State, to really try and better integrate the field work and empirical data on pollination networks with a lot of the mathematical modeling that has been done. And when it comes to networks, I think the modeling piece is very important, because we want to know, we want to be able to model the long-term dynamics, and experimentation in these systems is very challenging. But right now, these two approaches don't really talk to each other nearly enough. And so the three of us are really working to, again, integrate those two approaches in pollination networks. In particular, when I was at Emory, we did a whole series of behavioral studies using an artificial flower system in the lab and live pollinators. We have not built back this system up at UW, but I think that is something that will be coming down the line at some point, hopefully fairly soon. And then finally, we have also been, for several years now, working on the DNA metabarcoding of pollen. And that's a lot of fancy words that probably many of you have not heard of before, but really what we're doing is using the DNA that's inside of pollen grains to identify what those pollen grains are. um, often to the genus or to the species level. So what this allows us to do is really take the pollen load off of a single pollinator and then sequence it and then see what are the plants that that pollinator has visited. So that's been a fun thing to work on as well, and very directly related to this question of pollination networks. So with that, I'd like to thank a lot of people who helped make this work possible. And I'd say this is only a very um, uh, fraction of all of the folks that really deserve an acknowledgement here, but really lots and lots of people have contributed to this work, as well as many different funding sources have contributed to the work that my lab has done in various ways. With that, I'd like to really thank you for your attention